The amount of information and data that are becoming available to us is growing exponentially. As the amount of available data grows, so is our need to understand it. Statistics is the science of learning from data. This course will help you to develop the skills you need to analyze data and to communicate your findings. There won't be many formulas in the course. Rather, we will develop the key ideas of statistical thinking that are essential for learning from data. When you take this course, it is helpful if you have access to a spreadsheet, a software package or website that provides basic statistical functions, such as areas under the normal curve. But proficiency in a programming language is not necessary to master the contents and to complete the course. We will discuss the main tools for descriptive statistics which are essential for exploring data, with an emphasis on visualizing information. We will explain the important ideas about sampling and conducting experiments. Then we will look over some important rules of probability and discuss normal approximation and the central limit theorem. We will show you the important concepts and pitfalls of regression and how to do inference with confidence intervals and tests of hypotheses. You will learn the key ideas about computer-intensive inference with the Monte Carlo method and the bootstrap. We will show you how to analyze categorical data and discuss one-way analysis of variance. Finally, we will look at reproducibility, data snooping, and the multiple testing fallacy, and how to account for multiple comparisons. These issues have become particularly important in the era of big data. Broadly, there are three main reasons why statistical literacy is essential in data science. First, it provides the skills to assess whether the data are sufficient to answer the questions at hand. Second, it establishes a rigorous framework for quantifying uncertainty. And finally, it provides techniques for effectively communicating the findings of your analyses. This course equips you with the important tools in all of these areas. It is the statistical foundation on which the recent exciting advances in machine learning are built. I hope you will enjoy the course. Hello, my name is Günther Walter, and I would like to welcome you to the Introductory Statistics course. I'm a professor in the Department of Statistics at Stanford University, where I teach and do research. I regularly teach a variety of classes, from the introductory level for freshmen all the way to PhD level classes. I've been involved in developing the data science track in our Master of Science program, which the Department of Statistics runs jointly with the Institute for Computational and Mathematical Engineering. Together with some of my colleagues in the statistics department, I have also helped to create a data science course that is accessible to all students at Stanford, which incorporates many topics that you will find in this introductory statistics course. Research-wise, I have worked in a range of areas, from investigating puzzling behavior in solar neutrinos to extracting information from many measurements of single cells in high-throughput biological experiments, to developing new statistical methodology. One thing that I noticed when working in these diverse areas is that statistical questions come up everywhere, and that having a good grasp of the key ideas of statistical thinking can make a big difference. In our first module, we will look at descriptive statistics, that is, ways to summarize data with numbers and graphs. In particular, we will look at some important principles for visualizing information. This is important because most people prefer to look at figures rather than at numbers, so it's best to communicate information with figures whenever possible. Let's first look at an example that shows why descriptive statistics are so useful. In January 1986, the Space Shuttle Challenger broke apart shortly after liftoff. This accident was caused by a part that was not designed to fly at the unusually cold temperature of 29 degrees at launch. This was actually discussed by the engineers before the launch. Well, let's look at the data. Here's a list of the launch temperatures for the first 25 shuttle missions. And if you look at this long list of numbers, it's kind of difficult to see what's going on. However, if we make a simple graphic like this one, we see immediately that the temperature of 29 degrees at launch day is really far below all the other temperatures. So this makes clear why a simple graphic can be very useful. So while descriptive statistics has a number of uses, the two most important functions are to communicate information and to support reasoning about the data. Also, when the data are really large, then it becomes essential to use summaries because there's simply no other way to look at the data or to use the data. There are two ways to summarize data. One can either use numerical summaries, or one can summarize the data using pictures. For communicating information, it's usually best to use a graphical summary, and that's because people prefer to look at pictures rather than at numbers. Now, there are many ways to visualize data. Which one to choose will depend on the nature of the data and the goal of the visualization. And we will look at some examples now. First, let's look at the case where the data are qualitative. That is, the data are not numbers, but categories, such as colors. In that case, we would use a pie chart or a dot plot. On the left, there's a pie chart that shows the geographic origin of students at a university on the West Coast. The idea is that each slice of the pie is sliced according to the corresponding percentage. The right side shows the dot plot of the same data. The idea is that each horizontal line corresponds to one category. The position of the dot corresponds to the percentage. For example, if we look at Oregon, this dot corresponds roughly to 20%. Each of the two displays has some advantages and disadvantages. For example, if we want to compare two categories, it's easier to do that in a dot plot. If we look at the proportion of international students and the proportion of students from other US states, then it's very difficult to see which one is bigger. Whereas it's very easy to see that in the dot plot because it's much easier to judge the position of a horizontal line. On the other hand, the pie chart makes it easier to see what percentage of the total a category represents. For example, if we look at Oregon, then we see that this is roughly a little bit less than a quarter of the total pie. And this would be hard to see in the dot plot because we first would have to look down on the horizontal axis to see what's going on. Now, when the data themselves are quantitative, that is, they are numbers, then the convention is to put the data on the number line. The reason for this is that the ordering in the distance between the numbers conveys important information. The standard graph for displaying such data is the bar graph. It's essentially a dot plot put on its side. In this example, it shows the number of assignments that were completed by 22 students in a class. The histogram is very similar to a bar graph, but it allows to use blocks which have different width. For example, this histogram shows the ages of a number of people. The key point here is that the areas of the blocks are proportional to frequency. This means that the total area corresponds to 100%. For example, if we are interested in figuring out what percentage of people fall in the age group from 60 to 80, then we are interested in the area of this block. Now, just looking at that picture, we see that this area is probably around 1/7 of the total area. And so we can conclude that roughly 20% of the people fall into the range from 60 to 80. So the percentage falling into a block can be figured out without using a vertical scale, just using the fact that the total area has to be 100%. Now, sometimes it is useful to have a vertical scale. It's also called a density scale. And the reason for that is simply that the area of the block corresponds to percentage, and the area is computed by width times height. And since the width is given in years, the unit for the height needs to be percent per year in order for the units to work out. There are two kinds of information one can get out of an histogram. The first one is what's called density, which essentially means crowding. The height of the bar tells how many subjects there are for one unit on the horizontal scale. For example, if we look at the highest point in the histogram, which is around here, this corresponds to people of age 19. And looking at the vertical axis, we see that there are about 4% of all people in that age. In contrast, if we look at the block between 60 and 80, we see that the height of the block is maybe 0.007. That means about 0.7% of the subjects fall into each one-year group in that age range. The second piece of information one can get from the histogram is to compute percentages. Remember the rule which says the area corresponds to a percentage, and the area of a block is given by height times width. So, for example, if we want to figure out what percentage of people fall in the age range between 60 and 80, then the computation that we would make is we would look at the width of the box, which is 20 years, multiplied by the height, which is 0.7% per year, and we would find that there are 14% of subjects falling in that range. As we've seen before, there's another way to figure this one out. We can eyeball that this area comprises roughly 1/7 of the total area. So roughly 1/7 of the subjects fall in that range. I'm sure you've seen a bar graph and histogram before. The next one may be new. It's called the box plot. Sometimes it's also called the box and whisker plot. The idea is that it visualizes 5 key numbers of the data. In this example, we look at fuel consumption of 32 cars. It's given in miles per gallon. The lower whisker, which is the number here, shows the smallest number in the data. So we see it's roughly 10 miles per gallon. The upper whisker here shows the largest number in the data, which may be around 35 miles per gallon. And then there are 3 more numbers. We have the median. That's the number where half of the data are smaller and half are larger. So this might be around 19 miles per gallon. And there's the first quartile, which is this number here, around 15 miles per gallon. That's the number where a quarter of the data are smaller and 3/4 are larger. And finally, there's the third quartile, which is this number, which gives you the point where 3/4 of the data are smaller and 1/4 are larger. The box plot has less information than a histogram because it only shows 5 numbers of the data. But in turn, it takes up less space, and so one can use it to compare several data sets by putting them into the same graphic. The display below shows the same data that we had on the previous slide. But now the data are divided up into 3 groups, depending on the number of cylinders of each car. Each group gets its own box plot. By putting all 3 box plots in one graphic, we see that the miles per gallon are larger for cars that have fewer cylinders. A graphic like this is difficult to make with histograms. Also keep in mind that the histogram depends on the choice of the intervals that we select for the blocks. We don't have to deal with that when we use box plots. This graphic is an example of the principle of small multiples. We will talk about this later. Finally, let's look at data that consist of not just one number, but 2. That is, the data come as pairs. Those data are typically plotted in a coordinate system, which is called the scatter plot. The example here shows the years of education and the salaries for a number of people. For example, this person may have about 12 years of education and an income of roughly $15,000. The scatter plot is very useful to visualize the relationship between 2 variables. For example, we see that as education increases, income tends to go up. And moreover, we see that for many years of education, there seems to be a steep increase in income. The purpose of a statistic analysis is typically to compare the observed data to some kind of reference. Therefore, it's very useful to provide context in graphical displays. There's a nice explanation of this in the book The Visual Display of Quantitative Information by Edward Tufte. One way to provide context in the graphic is by using the principle of small multiples. We've already seen that the box plot is very useful for this. This graphic shows the temperatures that were recorded at a weather station over 50 years. It shows a box plot for each month. For example, this is the box plot for January. So it contains the data for the 50 recordings of January. This display allows to compare how the temperatures vary throughout the year. The reason why the principle of small multiples works so well is because the human brain is very good at decoding the information in these pictures. Here's another example showing small multiples. It shows the monthly unemployment rates for each state from January 1976 to April 2009. Each plot is a time series plot. That means on the horizontal axis we have time from 1976 to 2009, and on the vertical axis we have the unemployment rate. The line that's given in each graph shows how the unemployment rate changes over time. Moreover, there's an orange band which shows a normal unemployment rate of between 4 and 6%. By looking at these small multiples, one sees immediately that there's a sharp increase in the unemployment rate during the financial crisis in states such as California and Michigan, whereas things are not so bad in states such as Montana or South Dakota. Modern software provides a lot of freedom to design one's own graphics. There's a temptation to produce graphics that is flashy, but oftentimes that results actually in a poor outcome. This is a graphic that shows annual donations to 3 universities. Keep in mind that we are really comparing 3 numbers here. There's a number for Harvard, which is 683 million. A number for Stanford, which is 469 million, and a number for Columbia, which is 359 million. The graphic looks really interesting because it has 3-dimensional bars, but it is actually quite difficult to read. One reason for that is because we are looking at 3-dimensional blocks. So we are thinking we have to visualize volume, when in fact we are just comparing heights. But comparing heights is itself quite difficult because you are seeing a perspective plot here. The base of the 3 blocks is at different heights due to the perspective plot. Now, to help you see what's going on, there are grid lines in the background which allow you to refer the height of the block to the corresponding number. But as a consequence, it is really quite difficult to compare these 3 numbers. It would have been much better to put these 3 numbers in a dot plot, which makes it much easier to compare them. As another fun example, look at the Gettysburg PowerPoint presentation by Peter Norvig. Peter envisions what may have happened had Abe Lincoln fallen into the trap of using flashy graphics. So let's talk about numerical summary measures. If we want to summarize data using just one number, then we use the mean or the median. So everybody knows the mean, which is simply the average. The median is the number where half the data are larger and half are smaller. So it is in some way a midpoint. Let's look at that histogram which shows annual household incomes in the United States in 2010. Remember that in a histogram, area corresponds to percentage. So we can find the median by looking at the point where half of the area is below and half of the area is above. So here it is marked that the median is at about $50,000. And we can see that roughly half the area of the histogram is below 50,000 and half the area of the histogram is above 50,000. So now we know that both the mean and the median summarize the data, and the question is, which one should we use? It turns out that if the histogram is symmetric, then the mean and the median are actually the same. This histogram shows 100 of the first measurements that were made of the speed of light. The numbers on the horizontal axis show the speed measured after subtracting of 299,000 kilometers per second. We see that this histogram is roughly symmetric around this axis. Other data that result in symmetric histograms would be, for example, heights and weights of people. But when we look at the histogram of annual household incomes, we see that the histogram is far from symmetric. A histogram like this, where the right side is much longer than the left side, is called skewed to the right. In that case, the mean can be much larger than the median. In fact, for the annual household incomes, the mean household income is about $67,000, whereas we saw that the median is $50,000. For such a skewed histogram, it is better to use the median. Let's look at this hypothetical example where we look at 10 homes and we know that the median sales price is $1 million. Then we know that 5 homes sold for $1 million or more and 5 homes sold for less than $1 million. On the other hand, if we are told that the average sales price is $1 million, then it's not really clear what that means. If the average is $1 million, then we know that the sum of the 10 sales prices is 10 million. Now let's assume that one house sold for $8 million. Then we know that the total sales prices for the other 9 houses must be 2 million. The average sales price of the other 9 homes is those 2 million divided by 9, and that's about 200,000. So if we think that the average sales price of $1 million means that most houses sold for roughly $1 million, then we are very wrong in this case. So the lesson here is that if the histogram is very skewed, as is the case for incomes and house prices, then knowing the average doesn't really tell you much. It's much better to use the median in those cases. Let's go back to the histogram of annual household incomes. It says that the top 10% reported an income of $135,000. That means that 90% of households had incomes of less than $135,000 and 10% had incomes above $135,000. This point is called a 90th percentile. Furthermore, it says that the top 25% had household incomes above $85,000. This would be the 75th percentile, because 75% of households have incomes below that. The 75th percentile is also called the third quartile. That comes from the fact that if one divides the histogram in 4 equal parts, then one gets the 25th percentile, the 50th percentile, and the 75th percentile. So the 50th percentile is then the second quartile. And we already learned another name for that, which is the median. And the 25th percentile is the first quartile. Remember when we talked about graphical summaries, we talked about the box plot. The box plot shows 5 numbers about the data. The smallest number, which is given on the lower whisker, the first quartile up here, the median, the third quartile, and the largest number. The distance between the third quartile and the first quartile tells you how much the data are spread out. This has a special name. It's called the interquartile range. When it comes to measuring spread in the data, it's more common to use a different measure, namely the standard deviation. You've probably seen the notation x bar, which stands for the average of a list of numbers. Let's call them x1 to xn. Here's the formula for the standard deviation of these numbers. It's not that important to remember the formula, since calculators and computers can compute it for you. But let's just have a look what the interpretation of that formula is. What the standard deviation does is it looks at the difference of each number from its average, squares the difference, and then looks at the average of those squared differences. And finally, it takes the square root. Sometimes you see a formula that has n-1 instead of n in the denominator. Both of these formulas work roughly equally well. The mean and the standard deviation are often used to summarize data. The first one gives you a measure of center, and the second one gives a measure of spread. Both can be sensitive to a few large or small data, just as we saw in the example before. So if you're worried about that, for example, if the histogram is very skewed, then it may be better to use the median in the interquartile range. Of course, one problem is that many people don't know what the median in the interquartile range is, so you may have to explain that.
So my name is Andrew Radin. I'm the CEO and co-founder of Tuzar, and we are an artificial intelligence-based drug discovery company. By training, I'm a computer scientist. I did a bunch of startups prior to this, all in consumer space, all either large computer, large data science. I've worked on the systems that power a nation's telephone calls. I've built large-scale advertising networks that have served billions of ads and hit tens of millions of people. I've built large-scale video games on Facebook, all these sorts of things. And all these projects, while I was doing that as an entrepreneur, I kind of had a second life, if you will, working on social causes. So when my grandmother, when I was a child, my grandmother started a homeless shelter with some of her friends, which I've been involved with all the way to today as an adult. My wife and I have traveled to third world countries and helped people in need, and all this work had been very separate. And a number of years ago, I thought, I'd like to bring those things together in some way, and very interested in sort of helping the homeless in some meaningful way. And about that time, one of the largest, if not the largest, homeless encampment in the United States was close to my home. It's called the Jungle in San Jose. And I was thinking, I really should get involved and make a big impact. And my wife, a very wise woman, said, that's a social issue. That's not where your skills can be best applied. As a computer scientist, maybe you should think about how you can use data science to make a more meaningful impact in the world. And so, long story short, that's how I ended up, at Stanford, learning about how to use computer science, specifically in the field of medicine, and that got me to where I am today. Through the SCPD program at Stanford, there's a course offered in the bioinformatics track. And what that course is about is learning how other people have used data science and computer science to do some sort of medical discovery or move the field forward in some way. And in that course, you, at least when I took it, you learn a lot about what other people had done, methods they used, techniques they'd used to come up with some discovery. And one of the guest lectures was given by Marina Sirota. She's a professor now at UCSF. And she was talking about her work in using gene expression microarrays and differential expression between drug signatures with those who had and didn't have a disease to figure out which new medications could be potentially effective. And I just thought her work was absolutely fascinating because it came on the heels of another lecture where we were talking about this concept of integrative genomics, which is pulling in data from lots of different instrumentation, lots of different sort of views, if you will, about a particular problem, and how you can incorporate those different sort of data sets to figure out what's real and what's a false positive. And all this just kind of clicked together in my head with some work I had done in a couple previous startups, where I was thinking about this larger idea of incorporating this wide diversity of data sets and putting together a mechanism which will help explore those data sets and figure out what's real and what's not. right? And so that was the genesis of my project in that course, and then ultimately the technology behind the company today.
In this module, we will examine how data are produced. We will look at the main concepts for drawing samples and designing experiments. We will see when it is possible to attribute an observed effect to a treatment, such as a medical treatment, and when that's not possible. We will also look at some curious pitfalls. To explain what statistical inference is, let's look at a question you've probably heard a lot about in the news. What percentage of voters approve of the way the US president is handling his job? This question has been asked by polling companies since the 1930s. If you think about it, it's actually quite difficult to answer that question because you would have to ask each voter, and there are more than 250 million people of voting age in the US. It's a daunting task to ask all of these 250 million people, so this would seem to be an impossible question to answer. However, it's actually not that difficult to estimate that percentage. What a polling company does is it samples, say, 1,000 voters at random. Then it uses the approval percentage among those voters as an estimate for the approval percentage of all voters. So there are a few technical terms which we are going to use in the future. The population simply means the entire group of subjects about which we want information. In the previous example, this would be all US voters. The parameter is the quantity about the population which we are interested in. In the previous example, that would be the approval percentage among all US voters. The sample is the part of the population from which we collect information. That would be the 1,000 voters which were selected at random. Finally, a statistic or an estimate is the quantity we are interested in as it was measured in the sample. So that would be the approval percentage among the sampled voters only. An important point is that even a relatively small sample of 100 or 1,000 voters will produce an estimate that is quite close to the parameter, even if the population is very large, like the 250 million subjects in that case. We will see later why that's the case. This example shows why statistics can be such a powerful and useful tool. Now let's have a look how exactly we would sample these 1,000 voters at random. A simple way would be to select 1,000 voters from your neighborhood, say your hometown. This is called a sample of convenience, and it's not a good way to sample. The reason is that the voters of your hometown will likely be different from the population of US voters. For example, voters on the West Coast and on the East Coast tend to be more Democratic voters, whereas voters in the Midwest tend to vote more Republican. This will introduce what's called a bias. That means the sampling will favor a certain outcome. There are various kinds of biases. Let's look at the three most important ones. We already looked at selection bias. For example, a sample of convenience makes it more likely to sample certain subjects than others. Non-response bias means that the people who choose to respond to the question may be different from the non-responders. For example, parents are less likely to answer a survey request at 6 p.m. because they're busy with children and dinner. Finally, you've probably seen websites that allow you to rate businesses. The reviews you see there are not very representative because those reviews are more likely to come from customers who had very bad or very good experiences and therefore feel strongly about writing a review. This is called a voluntary response bias. The best way to avoid such biases is to use chance in some planned way in the sampling. The most well-known way of sampling is called a simple random sample. It means that subjects are selected at random without replacement. So if you sample 1,000 voters out of the US population, that means each sample of size 1,000 is equally likely to be selected. How would one choose a simple random sample? A popular way to do that is using random digit dialing. That means the computer will dial telephone numbers at random. That worked pretty well when every household had a landline. It's more complicated now with many people using cell phones instead, and the polling companies are figuring out ways to get around that. A more sophisticated way to sample is stratified random sampling. There, the population is divided into groups of similar objects called strata. For example, the US voters would be divided in urban voters, suburban voters, and rural voters. Then a simple random sample is chosen in each stratum and the results are combined. This sampling can result in a more precise estimate than simple random sampling. However, it's more complicated to execute. One thing to keep in mind is that since the sample is drawn at random, the estimate will be somewhat off from the true population parameter. This is called chance error. Also keep in mind that if we draw another sample, we will get a somewhat different chance error. So we can think of the estimate as being made up out of 3 parts. It should be somewhere close to the parameter. There may be a bias if our sampling is not done well. And finally, there's a chance error which is unavoidable. So while the chance error is unavoidable, it turns out we can make it small by taking a bigger sample size. Moreover, we can compute how large the chance error will be. So it's totally in our control to choose the sample size large enough such that the chance error is sufficiently small. And we will see later how to do that. Things are quite different for the bias, which is also called systematic error. If we increase the sample size, the bias will not get smaller. Rather, it will just repeat the error on a larger scale. Also, we typically don't know how large the bias is. So having a bias is a real problem. And that's the reason why we use chance in sampling in order to avoid a bias. You may have read in the news that people who eat red meat have higher rates of certain cancers than people who don't eat red meat. The statistical term for this is that there's an association between red meat consumption and cancer. But it's important to keep in mind that this does not mean that eating red meat causes cancer. This is because people who don't eat red meat are known to exercise more and drink less alcohol, and it could be the latter 2 issues that cause the difference in cancer rates. What we have here is called an observational study. It simply measures the outcomes of interest. In this case, it would measure the cancer rate among people who eat red meat and the cancer rate among people who do not eat red meat. Based on these measurements, one can say whether there's an association between these 2 factors. But, as we saw before, association does not mean causation, because there may be other factors. These are called confounding factors. Sometimes they have the name lurking variable. Now, what we really would like to know is whether eating red meat actually causes cancer. For that, we need an experiment. In an experiment, a treatment is actually assigned to people in one group but not in the other group. Treatment is a technical term. For example, if we have a new medication against high blood pressure, then the treatment would be to take this medication. The group that gets the treatment is called the treatment group, and the other group is called the control group. After we assign the treatment, we compare the outcomes in the 2 groups. We have already seen that confounders can be a problem. For that reason, the 2 groups should be similar. Of course, the treatment is different in the 2 groups. The best way to make sure that the 2 groups are similar is to assign the persons into the 2 groups at random. The way to think about the random assignment is that for each subject we toss a coin, and if it comes up heads, then the subject goes into the treatment group, and if it comes up tails, it will go in the control group. Another precaution we should take is that every subject in the control group gets a placebo. A placebo is very similar to the treatment, but it doesn't do anything. In the example where we want to test a new medication against high blood pressure, the treatment might consist of a pill. Then the placebo would also be a pill which doesn't have any medication in it. Why would we do that? It turns out that the idea of being treated may already have an effect by itself. This is called the placebo effect. Assigning a placebo to the control group makes sure that both groups are equally affected by this placebo effect. The third precaution is that the experiment should be double-blind. That means that neither the subjects nor the evaluators know which subjects are assigned to treatment and which are assigned to control. This is done to avoid biases. For example, if a doctor has to evaluate whether their medication helped, then the doctor might be inclined to give a positive assessment if the doctor knows that the patient has received the treatment. Let's briefly go back to the placebo effect. It sounds a bit mysterious. Indeed, the placebo effect is still not fully understood. It's somewhere on the boundary between biology and psychology, and it's probably one of the most interesting phenomena in science. If you are interested more in reading about the placebo effect, there's the accessible article, The Weird Power of the Placebo Effect Explained, by Brian Resnick. Remember that a good experiment requires that the subjects are assigned to treatment or control by a random process. The idea is that any confounders operate equally on both groups. Of course, since we are using a random assignment, there's a chance that one confounder is more prevalent in one group than the other. But here's the second key point. Because we use a random assignment, we can actually calculate the size of these chance effects when we evaluate the outcome of the experiment. We will see later how to do that.
So through my startups, I had been working with Paul Leaf. Paul was the former president of Electronic Arts, ran the show there for about 20 years before he started his own venture capital firm. He invested in some of my prior startups, and I was working for him doing diligence, some other companies. And so anyway, we were out to lunch, and he's sort of asking me, like, Andrew, what the heck you're doing? Like, you've done all these startups, and now you've gone back to school. Like, help me understand. And I told him about this project, and I said, you know, I've been working on this really cool thing. I'm using data science to find potential new drug treatments. I got one of the best grades in the class on that project. I'm really excited. I'm going to, you know, publish a paper on this, an academic paper. And Paul has, he's got this look when you do something really stupid. It's like his face does this thing, and he started to make that face, and I was a little baffled. Right? I just told him how I'm living my dream. I'm doing this thing that I'm so impassioned about. And he says to me, and he says, you know, Andrew, you just told me the story about how you want to change the world. Do you think you'll do that by writing a paper, or do you think you'll do that by filing a patent, starting a company, and bringing this technology into the world? And so that conversation just like a worm that dug into my brain, and I kind of couldn't get away from it because he was right. You know, it was more than just doing something to be impressive, and rather wanted to actually bring this technology to the world and do something meaningful with it. And that was the beginning of the transition from the academic world into creating a new startup around the technology.
In this module, we will discuss the basic rules of probability that we will need for our statistical methodology later on. It turns out that there are only a few rules that are essential. Even quite difficult problems in probability can be solved by applying these few rules in the right places. We will also look at some case studies that show how these simple rules can be used in surprising ways, for example, to build a spam filter for email. So what does it mean to talk about probability? Let's look at a data example. According to the National Center for Health Statistics, in 2015 there were about 4 million babies born in the US, and 48.8% of these newborns were girls. So then if we look at a baby that's born someday in 2015 and we ask ourselves, what are the chances that this baby is a girl? We would say, well, the probability of a baby being a girl is 48.8%. And we write this with capital P for probability, and then in brackets we write down the event we're interested in this case the newborn is a girl, and this equals 48.8%. So this example shows how the probability of an event is defined. It's defined as the proportion of times that this event occurs in many repetitions. So this definition requires that we look at a chance experiment that can be repeated many, many times. For example, if we toss a fair coin, we say the chances of getting heads is 50% and the chances of getting tails is also 50%. And that's something that can actually be checked. And believe it or not, some people did check it. In World War II, the South African mathematician John Carriage tossed a coin 10,000 times and found that 5,067 tosses resulted in heads. So that's roughly 50% of the tosses. This definition of probability as a long-run interpretation of frequencies can make it difficult to interpret probabilities for a single event. For example, the journalist David Leonard of the New York Times writes about this problem in his article, What I Was Wrong About This Year. Sometimes people use a different interpretation of probability. For example, if somebody says, the probability that my best friend calls today is 30%, then this statement clearly cannot be interpreted as a long-run frequency, because today, in the particular circumstances of today, happen only once. So what this person means is there's simply a subjective feeling of a 30% that this event will happen. So this is called a subjective probability. It's not based on experiments, and different people may assign different probabilities to the same event. Probabilities are always between 0 and 1. The key for computing probabilities is to apply a few rules repeatedly. We already saw that the probability that a newborn is a girl is 48.8%. So what's the probability that a newborn is a boy? Well, that's the opposite, so it's 1 minus 48.8%, which is 51.2%. And that's already our first rule. To write down rules, it's convenient to abbreviate events such as newborn is a girl with capital letters such as A. So our first rule, which is called the complement rule, says that the probability that A does not occur is 1 minus the probability of A. This is called the complement rule because A does not occur is also called the complement of A. Now let's look at a different chance experiment, namely rolling a die. A die has 6 sides, and each of them is equally likely. So each side has probability 1/6 of coming up. This is our second rule. It's called the rule for equally likely outcomes. If there are n possible outcomes, and they are equally likely, then the probability of an event A is the number of outcomes in A divided by n. In the next two rules, we learn how to compute probabilities of multiple events as those of the individual events. For the first one, we need a small definition. A and B are called mutually exclusive if they cannot occur at the same time. Let's look at an example. Suppose we roll a die twice. Let's call the event A to be the event where we see 1 spot on the first roll. B is the event where we see 6 spots on the first roll. And C is the event where we see 1 spot on the second roll. Then A and B are mutually exclusive because if we have 1 spot on the first roll, then we cannot have 6 spots on the first roll. But A and C are not mutually exclusive because if we have 1 spot on the first roll, then we can also have 1 spot on the second roll. So here is our third rule. It's called the addition rule. If the events A and B are mutually exclusive, then the probability of A or B happening is simply the sum of the probabilities of A and B. Our last rule involves two events being independent. That means that knowing that one occurs does not change the probability that the other occurs. In the above example, B and C are independent because B concerns the first rule and C concerns the second rule. And the outcome of the second rule doesn't depend on the outcome of the first rule. But the events A and B are not independent, because if we have a 1 on the first rule, then we cannot have a 6 on the first rule. So the outcome of A will affect the outcome of B. So the last rule is the multiplication rule. It says if A and B are independent, then the probability of A and B happening is simply the product of the probability of A and B. So if we look at these two rules, we see that the addition rule has an OR, and the multiplication rule has an AND. And in the addition rule we add, whereas in the multiplication rule we multiply. And likewise, the assumptions are different. For the addition rule, the events have to be mutually exclusive, and in the multiplication rule they have to be independent. This is what one has to keep in mind to keep these two rules apart. So let's do an example to apply these rules. Let's roll a die 3 times. We want to compute the probability that we get at least 1 6 in the 3 rolls. So how would we do that? So one idea is to write the event at least 1 6 as follows. It could mean 1 6 on the first roll, or 1 6 on the second roll, or 1 6 on the third roll. So now we have OR in between there. So that suggests we should try the addition rule. But there's a problem here because those 3 events are not mutually exclusive. Because if we get a 6 on the first roll, we can also get a 6 on the second roll. So we cannot use the addition rule, and we have to try a different approach. So let's try the complement rule. The probability of having at least 1 6 in 3 rolls is 1 minus the probability of its complement, which is no 6 in 3 rolls. So why am I doing this? No 6 in 3 rolls means no 6 in the first roll, and no 6 in the second roll, and no 6 in the third roll. So now you see there's an AND between the events. And that's just what I need, because I know the 3 rolls are independent. So I would like to use the multiplication rule. And for the multiplication rule, I have to use ANDs. So by using the complement rule first, I put the whole thing in a position where I can use the multiplication rule. So I can simply multiply those events, and then I can just plug in, because the probability of no 6 in the first roll is simply 5 6. And by the way, that's another application of the complement rule. So I get 1 minus 5 6 times 5 6 times 5 6, and if you plug it in your calculator, you see this is 41.4%. To explain conditional probability, let's look at spam email. Spam email contains certain words with a higher frequency than non-spam email, which is also called ham email. For example, the word money. And that's because one reason people send spam email is to get your money. The probability that the word money appears in spam email is 8%. And the way we write this is probability of money appears in email given, which is denoted by a vertical line, given that the email is spam is 8%. And the probability that money appears in an email given that the email is ham is only 1%. The conditional probability of B given A is now defined as the probability of B given A is the probability of A and B divided by the probability of A. And rearranging that already gives us what's called the general multiplication rule. The probability of A and B is the probability of A times the probability of B given A. So remember the multiplication rule we had earlier. In the special case where the events A and B are independent, the probability of A and B is simply the probability of A times the probability of B. So you see, in the special case of independence, the conditional probability of B given A is the same as the probability of B. And that makes sense because independence means that the outcome of B does not depend on A. So let's do some computations. It's known that the probability that any email is spam is about 20%. Let's compute the probability that a randomly chosen email has the word money in it. How would we do that? So remember what we know so far. We know that the probability that money appears given the email is spam is 8%, and the probability that money appears given that the email is ham is only 1%. But I want to know the probability of money, and I only have the conditional probability of money given spam and ham. So the key idea here is to artificially introduce that event whether we have spam or ham. The way this is done is we can write the event that money appears in the email as two events. The event that money appears in the email is spam, or the event that money appears in the email is ham. And now you see these two events are mutually exclusive because if the email is spam, it cannot be ham. And since there's an or in between these two, we can use the addition rule. The probability that money appears in the email is the probability that money appears in the email is spam, and the probability that money appears in the email is ham. And now I simply plug in the definition of conditional probability. The first term is the probability that the word money appears given the email is spam times the probability that the email is spam, and likewise for the second term. And all of these things I know and can simply plug in, and I get 2.4%. So these type of computations are actually used for spam filters. We already saw that the probability that the word money appears in an email, given that the email is spam, is 8%. But for a spam filter, we need to know a different conditional probability. We need to know the probability that the email is spam, given that money appears in the email. Because after all, we want to classify an email as spam if we think there's a high probability that it is spam. So we need that conditional probability rather than the first one. The formula for conditional probability says that the probability of B given A is the probability of A and B divided by the probability of A. But A and B is the same as B and A, so I can simply switch the roles of A and B in the numerator and write probability of B and A, and then I can use the same formula for conditional probability with the roles of A and B reversed. And what I get is this: probability of A given B times probability of B divided by probability of A. So what this simple calculation does for us is it computes a probability of B given A in terms of a probability of A given B. And that's exactly what we need for the above problem. We want to know the probability that the email is spam, given that the word money appears in the email. And applying that formula says that this equals the probability that the word money appears in the email, given that the email is spam, times the probability that the email is spam over the probability that money appears in the email. And we know all of these probabilities and can plug in, and we find 67%. So this rule has a special name. It's called Bayes' rule. This is the formula we derived above. And sometimes it turns out that the denominator, probability of A, is not directly given to us, and we have to compute it. And we do that just as we computed the probability of money on the previous slide. And if we do that, we get the expanded version of Bayes' formula, which looks like that. So the key to applying Bayes' rule is to always first try the simple version, and then if it turns out that we don't know the denominator, then we try the more complicated version. The key for Bayes' rule is it turns conditional probabilities of A given B into probabilities of B given A. The previous example where we computed probabilities in order to classify email as spam is a simple example of what's called a Bayesian analysis. The idea is as follows. Before looking at the email, we already know that about 20% of all email is spam. So there's already a prior probability of 20% that the email is spam. The idea now is to look at the evidence inside the email, such as certain keywords like money, and to improve on that prior probability using that evidence we find inside the email. So we did this using the Bayesian formula. And the way to think about this is that the Bayes' rule allows us to improve or update the prior probability into what's called the posterior probability that incorporates the information that we found inside the email. Here's another example where we will use Bayes' rule. Suppose that 1% of the population has a certain disease, and there's a test that tests for the presence of the disease. If an infected person is tested, then there's a 95% chance that the test is positive. But if the person is not infected, then there's still a 2% chance that the test gives an erroneous positive result. That's called a false positive because it's a positive result when in fact the person is not infected. Given that a person tests positive, what are now the chances that he has the disease? So let's look what probabilities we have. First, there's the 1%. That refers to the probability that a certain person has the disease. So we can write this down as the probability that the disease is present is 1%. Then we have the 95% chance that the test is positive if the person is infected. That's a conditional probability, the probability of a positive test given that the disease is present. Finally, there's the 2%. That refers to the probability of a positive outcome given that the disease is not present. What do we want to know? Well, we want to know the probability that the disease is present given that the test is positive. So now you see we are looking for a conditional probability, and what we have is a conditional probability with the roles of positive outcome and disease flipped around. So that immediately tells you that you want to use Bayes' rule, because Bayes' rule allows you to flip these two things around. When you plug in, this is what you get. So let's see whether we can get an answer from there right away. We know the probability of a positive test given that the disease is present, and we know the probability of the disease being present, but we do not know the probability of a positive test. So this is the case where we want to use the expanded version of Bayes' rule. Now we can simply plug in. Everything that's written there we know. For example, the probability of not having a disease is simply 99% by the complement rule. If you plug in, what you get is 32.4%. This is surprisingly low. After all, there's a positive test. The test seems to be quite good. 95% of the time when the disease is present, the test finds it. And only 2% of the time when the disease is not present will the test give an erroneous result. So how come then there's only a 32% chance that the disease is there if we have a positive test? The reason is that most of the population does not have the disease. Only 1% of the population has the disease, and the test finds most of those. But there's also a 2% chance of finding the disease for a non-infected person, and since there are so many people who are non-infected, that number is quite big. And that's why there's only a 32% chance that the disease is present among those tested positive. So here's a survey question that's quite tricky. What percentage of students have cheated during an exam in college? The problem here is that you cannot simply ask students whether they have cheated. Students may simply be too embarrassed to answer truthfully. So what can be done? This is being addressed with what's called randomization. It works as follows. We do a survey that first instructs students to toss a coin twice. If the student gets tails on the first toss, then the student has to answer question 1. Otherwise, the student has to answer question 2. Here are the two questions. Question 1 says, have you ever cheated on an exam in college? Question 2 is, did you get tails on the second toss? So why are we doing this? The answer we are going to get will be partly random. A yes answer could be due to the student answering question 1 and having cheated on an exam, or it could be due to the student answering question 2 and getting tails on the second toss. So a yes answer doesn't really tell us anything. And this should put the student at ease to answer truthfully. So if a yes answer doesn't tell us anything, then why does that work at all? So here's the key point. We cannot really tell what an individual yes means, but by looking at all the answers collectively, we can actually estimate the proportion of cheaters. Here's how it works. A yes answer can come from either question 1 or question 2. So we can divide up the event of a yes into 2 parts. This is the same reasoning that we've used before with a spam filter. So the probability of a yes answer could be a probability of a yes answer and question 1 was answered, plus the probability of a yes answer and question 2 was answered. And now I simply plug in the formulas for conditional probabilities, and I get this. What we are interested in is this probability. A yes answer on question 1, which is the question about cheating. So what we do is we solve for this probability and we get this expression. So let's see. We know the probability that question 2 was asked, that's just a half because it depended on the first coin toss. The probability that question 1 was asked is also a half. And the probability of a yes answer given question 2 was asked, that's also a half because question 2 simply asked whether you had tails on the second toss. So the only thing which we don't know is the probability that the answer will be yes. But this we can estimate from data. In one survey, 27 students answered yes and 30 answered no. So we can estimate the probability of a yes answer as 27 over 57, which is 47%. Now we plug in and we find that the probability of a yes answer, given question 1 was answered, is 44%. So we can estimate that about 44% of all students have cheated on an exam in college.
So I think a lot of the work we're doing is pretty unique in this field in that so we're certainly not the only folks using data science and drug discovery, but I would characterize a lot of those other efforts as going after either one type of data generation and sort of examining that data to see if there's some new insight there, or it's some sort of clever methodology to examine data that's already exists. And kind of that approach reminds me a bit about the children's story of the blind man and the elephant, right? When you're only inspecting sort of a small piece of the entire universe that represents a drug or a disease, you might look at that, elephant as that blind man and say, it's a rope or a spear rather than seeing the entire picture. And our view is it would be much more interesting to pull in the widest diversity of data you can get your hands on. And then across that sort of spectrum of information, help figure out what's a false positive, what's actually meaningful, help correlate a signal you see across these very different data sets, and use that to help focus in on what's the most likely drug treatment that's going to work for that disease and for those populations. For us, we probably fall more in the category of translational bioinformatics, which is saying we want to actually take data and translate that into some medical intervention in some way. And so, yeah, there's different disciplines. There's people who are just interested in raw knowledge and not necessarily trying to produce a diagnostic or a drug or sorts of things. So there's lots of different ways that technologies in this field can be applied.
We will now look at the empirical rule and normal approximation for data. This is an important technique in statistics because it is used in many statistical procedures. We will also discuss the binomial distribution and some basic ideas about random variables. It turns out that many data have histograms which have a very characteristic shape. Heights, weights, IQ scores, and many other data have a histogram which has a bell shape. The histogram that you see here shows the heights of 928 fathers that were collected by Sir Francis Galton in the 19th century. This is a typical example of a bell-shaped histogram. In such a case, one says that the data follow the normal curve. But keep in mind that some data have histograms that are quite different. We already saw that when we looked at incomes. It turns out that if the data follow the normal curve, then there's a rule of thumb which comes in handy in many situations. About 2/3 of the data fall within one standard deviation of the mean. This is called the empirical rule. There's a second part to the empirical rule, which says that about 95% of the data fall within 2 standard deviations of the mean. And in fact, about all of the data fall within 3 standard deviations of the mean. Let's look at an example. We saw that the height measurements of the fathers have a mean of 68.3 inches and a standard deviation of 1.8 inches. Now, the second part of the empirical rule says that 95% of the data fall within 2 standard deviations of the mean. That means if I go 2 standard deviations below the mean, that is, I take the mean minus 2 standard deviations, and I go 2 standard deviations above the mean, then I can tell that 95% of the height measurements fall between 64.7 inches and 71.9 inches. Let's see how the empirical rule looks like when we draw a histogram. Recall that in a histogram, percentages are given by areas. So if I draw a histogram that looks bell-shaped, then the mean is right in the middle. And if I go 1 standard deviation above the mean and 1 standard deviation below the mean, then the empirical rule says that 68% of the data fall between these two numbers. Now, can I say something about how many data are bigger than 1 standard deviation above the mean? Well, I know if 68% fall in the middle, that leaves 32% on the outside. And since the histogram is symmetric, it must be half on each side, so this would be roughly 16%. Now let's look at the second part of the empirical rule. The second part talks about going 2 standard deviation in each direction. So here's my mean. Here's the mean plus 2 standard deviations. And here's the mean minus 2 standard deviations. And then I know that 95% of the data fall between these two numbers. So that leaves me 5% outside, and in particular I get 2.5% on each side. A nice fact about the normal curve is that it is completely determined by the mean and the standard deviation. Once we know the mean and the standard deviation, then we know the whole histogram and we can compute percentages. To do that, we first standardize the data. Standardizing means that we take the data, in this case the height measurements, and we subtract off the mean and divide by the standard deviation. Oftentimes, the resulting standardized data is denoted by z. It's called a standardized value or z-score. Now keep in mind that z has no units. In this example where we looked at heights, heights come in inches, and the mean and the standard deviation all come in inches. But the z-scores have no units whatsoever. And that's simply because we have inches in the numerator and we have inches in the denominator, and so the inches cancel out. Z tells you how many standard deviations the measurement is above or below average. For example, if z equals 2, that means the height is 2 standard deviations above average. If z is negative 1.5, then we know that the height is 1.5 standard deviations below average. Once we standardize the data, they have mean 0 and standard deviation equal to 1. This is the whole point of the exercise. We already saw that father's heights follow the normal curve with a mean of 68.3 inches and a standard deviation of 1.8 inches. After we standardize, the standardized values follow what's called the standard normal curve, which has mean 0 and standard deviation 1. Here's a graph of the standard normal curve. You can plot this graph using the equation y equals 1 over square root 2 pi times e to minus a half x squared. Don't worry, we are not going to use that formula in this class. Normal approximation means to use the area under the normal curve to figure percentages. Let's look at an example. What percentage of fathers have heights between 67.4 inches and 71.9 inches? Remember that we know the mean and the standard deviation for those data, and that's really all we need to know. Normal approximation consists of 4 steps. In the first steps, we standardize the data. That means we take the endpoint 67.4 inches and we subtract off the mean, divide by standard deviation, and we get a standardized value of negative 0.5. Then we do the same thing with the other endpoint of 71.9 inches, and we get a z-score of 2. In the second step, we mark the area in the histogram we are interested in. 67.4 is somewhere here, and 71.9 is here. So we want to know the shaded area in that histogram. Once we standardize, we get a standard normal curve, and we've seen that the left endpoint gets standardized to negative 0.5, and the right endpoint gets standardized to 2. So now we have to find the shaded area under the standard normal curve. At this point, we use a computer or a table to look up that area. That requires us to do a small extra step, because typically software requires us to look up areas that come in a certain form. Typically, we can look up the area to the left of a given value. In our example, we were interested in the area between negative 0.5 and 2. Since software can give us only the value to the left of a number, we have to rewrite the whole thing accordingly. So what we are going to do is we write this as all of the area to the left of 2 and then we subtract off the area to the left of negative 0.5. Both of these areas can be looked up in computer programs. Once we look at these areas with software or in a table, we find that the first one is 97.7% and the second one is 30.9%. And so the area we're interested in is simply the difference, which is 66.8%. By now you've probably realized that the empirical rule is simply a special case of normal approximation. Remember, the empirical rule says that there's 95% of the data between the standardized values of -2 and 2. That leaves us 2.5% to the left of -2. So the total area to the left of 2 is 2.5% plus 95%. And that gives us 97.5%. So this rule of thumb is very close to the 97.7% that we got on the previous slide using software. Let's look at another example to see what normal approximation can do for us. Recall that the 30th percentile is the point where 30% of the data fall below. So if I draw a standard normal curve, then the 30th percentile would be the z-value where 30% of the data fall below. Using a table or software, one can find that z is -0.52. Now we have to figure out how to go from that z-value to the 30th percentile of the heights. That means simply doing the standardization backwards. Remember that the z-value is height minus mean divided by standard deviation. Now we know what z is, and we already know what the mean and the standard deviation is, so we can simply solve for height. All we have to do is plug in the numbers. Now there's a second way of doing this problem. Remember, z tells you how many standard deviations you are above or below average. So z equals -0.52 means that the height is 0.52 standard deviations below average. So the height would be the average minus 0.52 standard deviations, and then plugging in the numbers, we get 67.4 inches. So we saw earlier that for a newborn baby, there's about a 49% chance that it is a girl. Now, what are the chances that if we look at 3 newborns, we have 2 girls? So if we think back about the section on probability, we can compute this by simply listing all the possibilities. Remember, we called this total enumeration. So the probability that 2 out of 3 newborns are girls would be that we have a girl, another girl, and a boy, or we have a girl, a boy, and a girl, or a boy and a girl and a girl. Then we can simply add up these 3 terms because of the addition rule. Remember, for the addition rule, we required that the events are mutually exclusive, which they are. And finally, we can use the multiplication rule on each of these 3 probabilities because the 3 births are independent. So what we end up with is a term 3 for the 3 terms in the sum. And then we have 0.49 for the probability of a girl here, 0.49 for the probability of a girl, and 0.51 for the probability of a boy. The 3 counts the number of ways we can arrange 2 girls and 1 boy, and the product 0.49 times 0.49 times 0.51 is the same for all 3 terms. The calculation that we did on the previous slide was an example of what's called the binomial setting. We had 3 repetitions of an experiment, and these repetitions were independent. In our example, an experiment was simply giving birth to a child. Each of these experiments has two possible outcomes. In our example, these were the sex of the child, a girl or a boy. And in the binomial setting, these two outcomes are generically called success and failure. Success is typically the outcome we are interested in. So in our example, this would be that the newborn is a girl. And finally, the probability of success, which was 49%, was the same in each experiment. These three conditions define what's called the binomial setting. Now let's look at a little bit more challenging problem. What's the probability that 2 out of 5 newborns are girls? In principle, we can compute this just as before. So now we have to look at all the possibilities how we can arrange 2 girls among 5 newborns. And it turns out there are now 10 of these possibilities. I wrote down the first 3 here. You could have a girl, girl, boy, 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 or girl, boy, 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 and you get the idea. So the problem is that the number of these possibilities grows very quickly as n gets larger. But fortunately, there's a formula for computing those. That formula is given by the binomial coefficient, which counts the number of ways one can arrange k successes in n experiments. The formula reads n factorial, so that exclamation mark is pronounced as factorial. N factorial simply means you multiply up all the numbers up to n. For example, if we look at 3 factorial, that would be 1 times 2 times 3, which is 6. Then we have to divide by k factorial and n minus k factorial. As a special case, 0 factorial is always 1. In our previous example, we had n equals 3 births, and we wanted to know what are the chances to get k equal 2 girls. So we have to compute 3 factorial over 2 factorial times 1 factorial, which gives us 3. And that's exactly what we got when we did it by hand. Now we can use the binomial coefficient to compute the probability of k successes in n experiments. So here's the answer. This binomial coefficient, we already saw, it counts the number of ways to arrange k successes in n experiments. And then what we have is this term p to the power k times 1 minus p to the power n minus k, and that is simply the probability of having a particular pattern of k successes and n minus k failures. And that is because if you want to have k successes, it's p to the power k because they're independent, and each failure has probability 1 minus p, and since we have n minus k of them, we raise it to the power n minus k. This formula is called the binomial probability. So let's do an example about binomial probabilities. Suppose you play an online game 10 times. Each time there are 3 possible outcomes. You can win a big prize with probability 10%, you can win a small prize with probability 20%, and you win nothing with probability 70%. The question is, what is the probability to win 2 small prizes? Now it would seem that this is actually not the binomial setting because, after all, we have 3 possible outcomes. But if you look at the question, you see you're asked to compute the probability of 2 small prizes. It doesn't talk at all about big prizes. So this is, after all, an example of the binomial setting where success means you win a small prize and failure means anything else. That is, you win a big prize or you win nothing. So in the binomial setting, our probability becomes the probability of having 2 successes in 10 experiments. And for that, we can use the binomial formula. We have n is 10, so there's 10 factorial. Then k is 2, so there's 2 factorial. And 10 minus 2 is 8, so there's 8 factorial. Then the probability of success is 0.2. And that has to be raised to 2 because we want to have 2 successes. The probability of failure is 1 minus 0.2. And that has to be raised to the power 8 because 8 is 10 minus 2. So now we write 10 factorial as 10 times 9 times 8 factorial. Remember, 8 factorial means 8 times 7 times 6 and so on, so we can write it that way. 2 factorial is simply 2, and I leave the 8 factorial there. Then we keep the 0.2 to the power 2 times 0.8 to the power 8. And now you see you can cancel the 8 factorial, and you are left with 45 times 0.2 to the power 2 times 0.8 to the power 8. And if you plug this in your calculator, you see this is 30.2%. Now keep in mind that the outcomes of the 10 experiments are due to chance. So the number of successes you see are random. One set of 10 experiments might give you 4 successes. Another set might result in 7. For that reason, the number of successes is called a random variable. To make the notation easy, we simply write a letter for it, like x, just like in math. But we use capital letters to make clear that this is a random variable. So on the previous slide, we computed the probability of having 2 successes, and in that notation, that would simply be the probability that x is 2, and we found that's 30.2%. Since these were binomial probabilities, one says that the random variable x has the binomial distribution. Instead of computing the probabilities of all possible outcomes, we can simply visualize those probabilities with what's called a probability histogram. In the case of a binomial distribution with n equal 10 experiments and the probability of success of p equals 0.2, the probability histogram looks like this. The probability histogram is simply a bar graph where the height of the bar shows the probability of the corresponding outcome. For example, we saw that the probability of getting x equal 2 is roughly 30%, and that's the height of this bar. So here's the same probability histogram we had on the previous slide. Now remember, we talked about histograms before. In that case, we talked about histograms for data. And histograms of data give percentages for observed data. But keep in mind that the probability histogram has nothing to do with observed data. It's purely a theoretical construct. It shows probabilities, not data that have been actually observed. So if you look at the probability histogram for the binomial with 10 experiments and a probability of success of 0.2, you see that that histogram looks somewhat skewed. So there is a long right tail. But if we increase the number of experiments to 50, then you see that the corresponding probability histogram looks rather symmetric, and in fact it looks close to the normal curve. This suggests that we can use normal approximation to compute binomial probabilities, just as we used normal approximation before to compute frequencies for data. To use normal approximation for binomial probabilities, we need to standardize by subtracting of n times p and then dividing by square root n times p times 1 minus p. Let's look at an example of how to do that. A few slides ago, we were playing the online game where the probability of winning a small prize was 0.2. Now let's say we play this game 50 times. What is the probability of getting at most 12 small prizes? We already know that this is the binomial setting, so we could use binomial probabilities. But this is rather tedious work. We would have to compute the probabilities of having 0 successes, plus the probability of having 1 success, plus the probability of having 2 successes, and so on, all the way up to 12. On the other hand, if we use normal approximation, then this is a rather quick computation. Remember that the probability histogram of the binomial distribution with n equals 50 and p equals 0.2 looks roughly like a normal curve which is centered at around 10. Now we want to compute the probability of at most 12 successes. So here's 12, and that means we want to compute the shaded area to the left of 12. So we know in order to do normal approximation, we first have to standardize. To standardize, we have to subtract off np, so we take 12, we subtract off n times p, and we divide by square root n times p times 1 minus p. So we take 12 minus 10 divided by 2.83, and that gives us 0.71. So after we standardize, we get a standard normal curve, which is centered around 0, and the standardized value for 12 is 0.71. And so we have to find the area under the standard normal curve to the left of 0.71. Plugging this into software, we see that this area is roughly 76%. And that's going to be the answer to our question. Remember that when we talked about sampling, we noted that the poll typically selects subjects in a simple random sample, and that means sampling without replacement. If one is sampling without replacement, then this is not the binomial setting. For example, the probability of success p changes after a subject has been removed. But if the population we sample from is much larger than the sample, then sampling with replacement is about the same as sampling without replacement. So in that case, even if we take a simple random sample, that is, we sample without replacement, the number of successes will be approximately binomial. And that means we will be able to use normal approximation.
In life sciences in general, data sciences is being applied across the spectrum, right? So we're working in the earliest stages of drug discovery, but there's certainly work that data science can do to help optimize clinical trials. So part of having an effective drug therapy is knowing in advance whether or not that medication will work for you. And maybe you've had this experience, right? You go to your doctor, you're not feeling well, you're given a prescription, and then the doctor says, and if this doesn't work, right, come back in a couple weeks and we'll try something different. And so part of what's going on there is that your genetic makeup as an individual and sort of you as a biological entity, you might react differently to a medication than someone else. And so some of the other work that data science can help with is some of those companion diagnostics, right, to figure out which treatments would be most effective with which people. This is not work that we do as a company, but these are all opportunities where artificial intelligence data science helps, make people healthier as a whole.
The main topics in this module are the law of large numbers and the central limit theorem. The name central limit theorem comes from the central place that this theorem has in statistics. So it's important. But it's also important to understand that in every statistical analysis, there are several histograms floating around. The probability histogram which generates the data, the histogram of the observed data, and the probability histogram of an estimator. It's important to understand these and to keep them apart, and that's another goal of this module. So let's have another look at the distinction between a parameter and a statistic. What's the average height of adult men in the United States? That's quite difficult to get a handle on because there are 120 million adult men. But it turns out that one can estimate this number quite well with a relatively small sample. In this section, we'll see why that is. Remember that the population in this case is all adult men in the US, and there are about 120 million of them. A parameter is something about the population we are interested in. For example, the population average, which we call mu, or the population standard deviation, which we call sigma. A statistic or an estimate is this quantity as it's measured in the sample. That could be the sample average x bar or the sample standard deviation s. So the idea is that we will use the statistic from the sample to estimate the parameter of the population. Now, if we draw an adult male at random from the population, then we expect his height to be around the population average mu, give or take about 1 standard deviation sigma. After all, the standard deviation sigma measures the spread of the population, and most observations are around 1 standard deviation away from the mean mu. We say that the expected value of one random draw is the population average mu. Now, how about the average of n draws, which we call x bar with a subscript n? So the subscript stands for the sample size. So it turns out that the expected value of the sample average is again the population average mu. But keep in mind that the sample average is actually random, because sampling is a random process. That means that x bar won't be exactly equal to the population mean, which is actually 69.3 inches. For example, we might get x bar equal to 70.1 inches. And if we take another sample of size n, we might get x bar equal to 69.1 inches. So then the question is, how far off from the population mean will the sample mean be? This is determined by the standard error, or SE. This is a very important quantity in statistics, and it tells you roughly how far off the statistic will be from an expected value. The standard error of a statistic is used for all kinds of statistical procedures. The standard error of a statistic plays the same role that the standard deviation sigma plays for one observation drawn at random, because it's a give or take number that shows how far off we expect the statistic to be from the mean. So here's one of the key facts for statistical inference. It's called the square root law. It says that the standard error of the sample average equals sigma divided by square root sample size. Why is the square root law so important? There are actually two reasons for that. First, it shows that the standard error becomes smaller if we use a larger sample size n. After all, there's a square root n in the denominator. We can actually use that formula to determine what sample size is required to get a desired accuracy for our standard error. The second point is that the formula does not depend on the size of the population, only on the size of the sample. This is the reason why statistics works in polls. After all, remember our previous example where we looked at 140 million adult men. It doesn't really matter that we look at 140 million. If we take a sample of size 1,000, we get a certain standard error no matter how large the population is. Sometimes we are interested in the sum of the n draws rather than the average. Now we get the average by taking the sum and dividing by n, and so we get the sum back by taking n times the average. So it turns out that the formulas for the expected value and the standard error of the sum can simply be obtained by multiplying the formulas for the sample mean by n. The important thing to keep in mind here is that the standard error for the sum actually increases at a rate of square root n. So while the standard error of the average goes down, the standard error of the sum goes up. Another important statistic that comes up all the time are percentages. We already looked at approval ratings for US presidents. That's a question that was introduced by George Gallup in the late 1930s, and it's asked regularly every week or so. That poll tries to figure out what percentage of likely voters approve of the way that the US president is handling his job. Looking closely at this, we see that the percentage of likely voters is actually an average. It's helpful to look at this in some more detail because it comes up all the time. It's called a framework for counting and classifying. In this example, the population consists of all likely voters, which are about 140 million adults. Each of these likely voters falls into one of two categories. Either they approve of the president's handling of the job or they don't. What we do now is we put the label 1 on each likely voter who approves and 0 on each who doesn't approve. The reason we do that is because then the number of likely voters who approve equals the sum of all 140 million labels. To see why that is, let's look at a simple example with 5 voters. Suppose the first voter approves, so we give him a label 1. The second voter doesn't, so he gets a label 0. The third voter doesn't, gets a label 0. The next voter approves, gets a label 1, and the last voter doesn't and get a label 0. So if we look at the sum of the labels, then that will be 2. And indeed, 2 of the 5 voters approved of the president's handling of his job. So by putting 0s and 1s on the labels, the sum of the labels simply counts how many approve. And likewise, one sees that the percentage of likely voters who approve is simply the percentage of 1s among all the labels. So if we introduce labels, then sampling n likely voters means that the number of voters in the sample who are approving is simply the sum of the n draws. Likewise, the percentage of voters approving is the percentage of 1s. And that's then simply the sample average. And we can multiply by 100% just to convert the whole thing into percentages. So the bottom line is that percentages are simply averages after introducing 0 and 1 labels. That means we can simply use the formulas for averages, and we find that the expected value of the percentage of 1s is mu times 100%, and the standard error for the percentage of 1s is sigma over square root n times 100%, where mu is the population average and sigma is the standard deviation of the population of 0s and 1s. So now we have a number of formulas for the average, for the sum, and for the percentages. But really they all look quite similar. In the end, it comes down to sigma over square root n. But remember that all of these formulas are actually for sampling with replacement. But polls are simple random samples, so those are samples without replacement. Now we already saw that in the case of sampling without replacement, where the sample size is much smaller than the size of the population, these two things are roughly the same, so all of these formulas are still approximately true. In fact, it turns out the formulas for the expected values are exactly true, even for sampling without replacement. And for the standard error, it's only approximately true, and there are explicit formulas to correct for sampling without replacement. All of these formulas are also true if we don't draw from a population, but we simulate data. Simulating data means we generate data according to a probability histogram, for example, by using a computer. Now, what are mu and sigma in that case? Remember, when we draw from a population, mu is simply the population average, and sigma is the population standard deviation. If we simulate a random variable x that has k possible outcomes, x1 to xk, then the formulas for mu and sigma square are given here. These formulas actually also apply to the case of sampling from a population, because if we sample from a population with k possible subjects, then the probability of picking one subject is simply 1 over k, and the whole thing becomes the population mean. Likewise for sigma square, where this formula simply turns into the formula for the population standard deviation squared. In the case where the random variable x has infinitely many possible outcomes, such as when x follows the normal curve, there are analogous formulas, but we don't need these for the rest of the course. So now let's look at an example that applies the square root law. We toss a fair coin 100 times. How many tails do we expect to see, and what's the give or take number? This is an example where we count. We count how many tails we're going to see in 100 tosses. So we will introduce labels where a 1 stands for tails and a 0 stands for heads. Then we simulate these 100 tosses either by really tossing a coin 100 times or by using a computer, and in each case we have the probability of getting a 1 is a half, and the probability of getting a 0 is also a half. Because we have 0 and 1 labels, we find that the number of tails in the 100 tosses equals the sum of the 100 draws. So then we can use our formula for the expected value of the sum, and it says it's the sample size, which is 100, times mu. Now the formula for mu on the previous slide says it's the sum over all outcomes times their probabilities, and that is a half. So the expected value of the sum is 50, and that would be our answer. We expect to see 50 tails. Now the give or take number is the standard error. So we have to look at the standard error of the sum, and the formula says this is square root sample size times sigma. So we have to figure out what sigma is. The formula for sigma square is take each outcome, subtract of the mean, square it, times the probability, which is a half, and then we have to do that for the second outcome. And we find this is 1/4. So now, since we are looking at sigma, not sigma squared, we get 10 times 1/2 instead of 1/4, and that is 5. So our answer would be that we expect to see 50 tails, give or take 5. By the way, if we did this for the percentage of tails, then the formula for the percentage says we take sigma over square root sample size, and that is 0.05 or 5%. So in terms of percentages, our answer would be we expect to see 50% tails, give or take 5 percentage points. Let's look again at the previous example of tossing a coin 100 times. If we are interested in the number of tails, there are the following possible outcomes. We could get 0 tails, or 1 or 2, up to 100. Now, how likely is each of these outcomes? We know that this is the binomial distribution with n equal 100 and p equal 1/2, because if we call a coin landing tails a success, then the number of tails is simply the number of successes in 100 experiments. So if we are interested in the statistic that counts the number of tails, then this statistic is a random variable whose probability histogram is given by the binomial distribution. This is called the sampling distribution of the statistic. The reason why we use a sampling distribution is because it provides more detailed information about the chance properties of the statistic than the summary numbers given by the expected value and the standard error alone. The technical term distribution comes up in many different ways. It's helpful to step back and see what's happening here. When we tossed a coin 100 times, we simulated data by drawing from the probability histogram. That probability histogram says we can get heads with probability 1/2 and tails with probability 1/2. So remember that the probability histogram is a theoretical construct. It simply tells us what the chances are for simulating. After we simulate 100 tosses, we have actual data. For example, we could end up with 47 heads and 53 tails. Then we can make a histogram of those data, and the histogram looks like this one. So you see that this histogram looks quite similar to the probability histogram in the first step, but it's a little bit different because the 100 tosses involve a chance process. Finally, if we are interested in a statistic, such as the number of tails in 100 tosses, then we get a third histogram, which gives the sampling distribution of that statistic. In this case, it's a binomial distribution, and you see that this histogram is quite different from the first two. Later on, when we do statistical inference, it's easy to mix up these histograms, so it's really important to carefully think through what's going on there. The square root law means that the standard error of the sample mean goes down as the sample size increases. This means that if the sample size is large enough, then the sample mean will be close to its expected value mu. This is the famous law of large numbers. So keep in mind that the law of large numbers applies only for averages and percentages. But it doesn't apply for sums because we saw that the standard error of the sum goes up as the sample size increases. Also, we need to be sampling with replacement from a population or simulating the data with a computer from a probability histogram. More sophisticated versions of the law of large numbers, in fact, state that the whole empirical histogram of the data, which is the second histogram among the three we considered previously, will be close to the probability histogram if the sample size is large enough. Now let's go back to the online game, which we considered earlier, where you win a small prize with probability 0.2. We gamble n times and count the number of small prizes, and we called this a random variable x. At that time, we saw that x has a binomial distribution with n and p equals 0.2. Here are some probability histograms for that binomial distribution. If n equals 1, we see that it's a very much a skewed histogram, and for n equals 10, it still has a long right tail. But for n equals 50, the whole thing looks pretty much like a normal curve. This is an example of the famous central limit theorem. It says that when we sample with replacement and n is large, then the sampling distribution of the sample average, or of the sum, or of the percentage, approximately follows the normal curve. That means we can use normal approximation to compute probabilities. To standardize, we subtract off the expected value of the statistic and then divide by its standard error. The name central limit theorem comes from the fact that it has a central place in statistical theory. The reason why the central limit theorem is so important is that it shows that the statistic has a normal distribution no matter what the population histogram is. Let's look back at the distribution of annual household incomes in the United States. We saw that this distribution is very much skewed to the right, so it's very far from normal. The mean of the household incomes was $67,000 and the standard deviation was $38,000. So if we sample n incomes at random among all the households, then we know that the sample average follows the normal curve, even though the histogram of the incomes itself is far from normal. And to do normal approximation, we need to subtract off the expected value of the statistic, which in this case is the average of all incomes, namely 67,000. And then we have to divide by the standard error of the statistic, and we know by the square root law that one is 38,000 over square root sample size. For example, let's take 100 incomes. If n equals 100, then square root n is 10, and so the standard error is equal to $3,800. The central limit theorem says that the sample mean follows the normal curve centered at $67,000 with a standard error of $3,800. So the empirical rule says if we go one standard error above the average, then there's a 16% chance to fall above that number. And one standard error above average is precisely that 70,800 we were looking at. Let's go back to our example about online gambling. We did n gambles and looked at the number of small prizes, which we called x. Remember, when counting things, we use labels which have zeros and 1 on them. In that case, everything a small prize comes up, we give it a label 1, and 0 goes to everything else. That means that the number of small prizes simply equals the sum of these labels. And since we are now looking at the sum, we can use the central limit theorem. To apply normal approximation, we need to find the mean and the standard error of x. So remember, when we looked at simulations, we had formulas for the expected value and the standard error. And if you plug in, you find that in this case, mu equals p, and sigma is square root p times 1 minus p. That was the formula for one gamble. Now we are looking at the sum of n gambles, and we had formulas for the expected value and standard error, And these formulas show that the expected value of the sum x equals n times p, and the standard error is square root n times p times 1 minus p. Those are the numbers we use when we standardize x. And remember, we found the same thing when we talked about the binomial distribution. Let's summarize the main requirements for the central limit theorem to apply. First, we need to sample with replacement, or we need to simulate independent random variables from the same distribution. It turns out there are actually also central limit theorems for sampling without replacement. And in any case, we know that if the sample size is much smaller than the population size, then sampling without replacement is about the same as sampling with replacement, and so everything works out. The second important requirement is that the statistic we are looking at must be a sum. And remember that averages and percentages are essentially sums after multiplying. Finally, the sample size has to be large enough. The rule of thumb is that the more skewed the underlying population histogram is, the larger the required sample size n. It turns out that if there's no strong skewness, then a sample size of at least 15 will be sufficient. For stronger skewness, we might need a sample size of at least 40. Regression is arguably the most important statistical technique. It is extremely versatile and can be used for problems where at first sight one might not have expected that regression is useful. We will discuss the main ideas for inference in regression and how to do regression diagnostics. We will also look at some pitfalls. The histogram on the top left shows the heights of 928 suns. The average of their heights is 68.1 inches. Now suppose we pick one of these suns at random, and the task is to predict his height. In fact, what one typically finds in statistics is taking the average is in some sense the best predictor. Now suppose we have more information. Not only do we have a histogram of the heights for the suns, but we also know the heights of their fathers. The heights of these 928 father-son pairs are given in the scatter plot on the left. Again, we have to predict the height of a son, but now suppose we know that the father is 72 inches tall. Clearly, this additional information should help us somehow in our prediction. The reason for this is that if we know that the father is 72 inches tall, then we know that we are somewhere among this group of fathers and sons. And we know that for this group, the heights of the sons are on the higher end. What this means is that the additional information of knowing the father's height should allow us to come up with a better predictor. And that is precisely what a regression does for us. Before we can do regression, we need to know about the correlation coefficient. Remember that the scatter plot is very useful in visualizing the relationship between two quantitative variables. For example, in the left scatter plot, which shows education and income, we see that there seems to be an upward direction of the scatter plot. That is also true for the scatter plot that shows the heights of the fathers and their sons on the right. However, these two relationships are somewhat different. If we look at the relationship of income and education, we see that there seems to be kind of an upward sloping curve which the scatter follows. On the other hand, if we look at the heights, we see that the scatter roughly follows a line. Finally, the last thing we can get out of a scatter plot is the strength of the relationship. That means how closely do the points follow the form. In the example of the heights, we see the scatter is quite wide around the line, whereas in the example of the incomes, it's more closely around the curve. In the case where the scatter clusters around a line, it's very useful to summarize that clustering with a correlation coefficient R. It's not worth to remember the formula for R, but let's look at what's going on there. You see that we are looking at the standardized values of the x's and the standardized values of the y's. and then we multiply them together and take the average over all the observations. Now you see that this product here is positive if both x is above x bar and y is above y bar, and it can also be positive if x is below x bar and y is also below y bar. So this contribution will be positive if both x and y vary in the same direction from their averages, and it will be negative if they vary in opposite directions. So the idea of the correlation coefficient is that the correlation coefficient should be positive if the scatter slopes upward, and it's negative if it slopes downward. So if we have pairs of data and we see that their scatter follows a linear form, then we can summarize these data by the average of the x's, the standard deviation of the x's, the average of the y's, the standard deviations of the y's, and finally the correlation coefficient r, which tells us something about the relationship between x and y. When we plot these pairs x and y, then we use always the convention that whatever goes on the horizontal axis is called the explanatory variable or predictor, and the one which goes on the vertical axis is called the response variable. It turns out that the correlation coefficient r is always between -1 and 1. The idea is that the sign of r gives the direction of the association, whether it slopes up or down, and the absolute value of r gives its strength. Here are a number of examples. In the leftmost case, we have r equal to -0.9. Because r is negative, the whole scatter slopes downward. 0.9 means that the scatter is tightly clustered around a line. In the second example, we have r equals -0.6. Again, the scatter is sloping downward, but 0.6 is much closer to 0, so we see that the scatter is much more spread out. Finally, if r equals to 0, then there's no perceptible upwards or downwards trend. Now let's go to positive correlation coefficients. If r equals 0.2, we get an upward scatter, which is rather loose. Finally, if r equals 1, the scatter slopes upward and everything falls perfectly on a line. These examples give you an idea how to think about the correlation coefficient. Keep in mind that the correlation coefficient r comes without units, and that's because both x and y were standardized when we computed r. It also turns out that r is not affected by changing the center or the scale of either variable. For example, if you compute the correlation coefficient between height and weight, then it doesn't matter whether you measure weight in pounds or in kilograms. It's important to keep in mind, however, that the correlation coefficient is only useful for measuring linear association. Look at this example here. Clearly, there's a very strong association between the two variables. You see a tightly clustered scatter around a curve. However, if you compute the correlation coefficient, you see r equals to 0. R equal to 0 suggests that there's actually no linear association between these two variables. That's true, but of course it misses the point in this case. The bottom line is that r is really only useful when we look at linear scatters. Finally, it's tempting to see a large correlation coefficient and conclude that there must be some type of causal correlationship between the two variables. But the example on the bottom left shows that this is just not true. That scatter plot shows shoe sizes as well as the score on a reading test for 100 school children. Clearly, there's a very strong association between these two variables. But we would agree that shoe size has no causal effect on reading ability whatsoever. Rather, what's going on here is that there's a third variable, namely the age of the school children, which determines both the shoe size and the reading ability. So what's going on here is that correlation does not mean causation, and we've talked about that before. So how does regression work? Let's look at this scatter. It shows the age and the percent of body fat for a number of people. This scatter appears roughly linear, so this is a case where we might use regression. The idea is that we want to summarize the scatter by a line. This line should look something like this one, and the question is, how do we get it? Recall that the equation of a line looks like this. You have an intercept A plus a slope times the argument x. So if we plug in the ith x variable, then the equation of a line gives us a point which we call yi hat. The idea of finding the line is we want to look at those values of a and b that minimize the difference between the point on the line yi hat and the true observed value yi. One way to do that is to look at the difference between yi and yi hat, square it up and sum it over all the observations, and then we want to find a and b to minimize that sum. Minimizing that sum in a and b can be done either by calculus or simply using software on the computer. The whole idea is called the method of least squares. It turns out that the solutions involve all the summary quantities that we looked at before. The slope equals the correlation coefficient times the ratio of the two standard deviations, and the intercept involves the means as well as the slope previously derived. This line is called the regression line. Now there's another interpretation of the regression line. It computes the average value of y when the first coordinate is near x. Remember, the idea in statistics is that an average is oftentimes a best predictor. So by computing averages of y values where the first coordinate is near x, we finesse that argument somewhat to incorporate the information given to us in terms of x. And the idea would be that this is a better predictor of y than simply taking the average of all the y's. Now the main use of regression is to predict y from x, and we can simply do that by plugging in x into the equation of our regression line, which gives us the predicted value y hat. Now from the formulas on the previous slide you can see that if we predict that x equal the average of all the x's, then this prediction will simply produce the average of the y's. But the formula for the slope means that if we take x to be one standard deviation above the average, then the predicted y value is only r times one standard deviation above the average of the y's. Now r is typically smaller than 1 and larger than -1, and so this means that prediction is towards the mean, in a sense that the predicted value y hat is fewer standard deviations away from its mean than x is from the mean of the axis. Let's look at this scatter plot that shows scores on midterms and final exams for a number of students. The midterm scores are on the horizontal axis and the final exam scores are on the vertical axis. You see that the regression line clearly shows regression towards the mean. For example, if you look at students who did very well on the midterm, it would be roughly those, very much on the right-hand side. You see that their final exam scores, which are given on the vertical axis, are somewhat below the top scores on the final exam. So regression towards the mean actually makes sense. We predict that the top scorers on the midterm will score somewhat below the top on the final exam. Likewise, if you look at the worst scorers on the midterm, it would be roughly those. But those students did actually not score the worst on the final, because the worst scores on the finals are below there. Again, we would predict correctly that the final exam scores should not be as far away from the average as the midterm scores were. This is called regression to the mean, or the regression effect. One way to remember that is to look at a test-retest situation just as the example where we previously looked at the midterm and final exam scores of students. In such a test-retest situation, the top group on the test will drop down somewhat on the retest and the bottom group moves up. Why would that be? Here's a very heuristic explanation. To score among the very top on the midterm requires two things. First of all, the student has to be prepared very well, and there's probably also some luck involved. For example, the students may be very well prepared for the questions that happen to be on the midterm. Another element of luck might be that the student had a very good sleep the night before. Now, when we go to the retest situation, like the final exam, then some of the students may not have that luck anymore in their favor. And that's the reason why we expect this group to fall back a little bit. The same thing applies to the bottom group. The worst score is probably due to some degree to some bad luck, and on the retest situation there's hope that this bad luck is not there anymore. It's important to realize that this is simply an effect of the scatter around the line. Sometimes people look at that effect and conclude that there must be something else going on. For example, the conclusion might be that the top scorers on the midterm slackened off and did worse on the final. Such an erroneous conclusion is called the regression fallacy. Now let's do a prediction. Regression says that if we are given x, then we use the regression line to predict y. That means we compute the regression line, plug in x, and see what predictor y hat we get. As noted before, in order to compute the regression line, all we need to have are the 5 summary numbers of these statistics. Computing the regression line can be done very quickly in software. For example, the command lm in the computer language R will do that for you. But it turns out you can actually do it rather quickly by hand. Let's look at an example. Let's say the average midterm score was 49.5, the average final score was 69.1, the standard deviation on the midterm was 10.2, the standard deviation on the final 11.8, and the correlation coefficient R was 0.67. Now suppose somebody tells you a student scored 41 on the midterm, and you have to predict the final exam score of that student. Remember, if we wouldn't have that information about the midterm score, then the best predictor for the final would simply be the average of 69.1. But regression gives us a tool to come up with a better predictor by incorporating the additional information that the student scored 41 on the midterm. Here is how we can do that regression by hand very quickly. First, note that 41 is 8.5 below the average. Here, average refers to the average of the midterm scores, which is 49.5. Now we standardize this. 8.5 below average means 0.83 standard deviations below average. Looking at the formula for the slope of the regression line, we predict the final exam score to be only R times 0.83 standard deviation below average. So now we can simply plug in the numbers. We take the average for the final exam. We subtract off, because we are below average, so we have to subtract off R, which is 0.67, times 0.83, times the standard deviation for the final exam scores, which is 11.8, and we arrive at 62.5. This will be our prediction that we got from regression. Now let's turn the prediction around. Suppose I told you that a student scored 89 on the final, and your task is to predict the midterm score of that student. Here's one important thing about regression. When you predict x from y, it is a mistake to use the regression line you got from predicting y on x and simply solve for x. It's somewhat tempting, but it's the wrong thing to do. The reason why it's wrong is because there are two regression lines. There's one regression line for predicting y on x, and there's a different regression line for predicting x on y. These two lines will typically be very different. To avoid the confusion between these two lines, it's always best the predictor on the x-axis and whatever you want to predict on the y-axis. And then you simply do what we did before. So in this case, that would mean we look at an x-axis which corresponds to the final exam score, and a y-axis which shows us the midterm scores. That's because the final exam score is the thing we base our regression on. That is, the final exam score is the predictor. So we know the average of the final exam scores was 69.1. The average on the midterms was 49.5. And the information we are given is that the final exam score is 89. So it's above average. We know that the regression line goes through the points that are the average on the horizontal axis and the average on the vertical axis, and it slopes upward because r is positive. So the regression line looks somewhat like this. Since we base our regression on a final exam score of 89, we expect to end up somewhere above average for midterm. Exactly how much above average is this calculation which we just did before. So we say that 89 is 9.9 above average. So if we standardize, we get 9.9 divided by the standard deviation of 11.8 equals 0.84 standard deviations above average. Therefore, our prediction for the midterm will be also above average, and it will be not 0.84 standard deviation above average, but r times 0.84 standard deviations above average. And now you can just plug in. We take the average for the midterms, which is 49.5. Since we are above average, we add r, which is 0.67, times 0.84, times the standard deviation, which is 10.2, and that gives us 55.2. This will be our prediction for the midterm score. Now keep in mind, if r were negative, then the line would slope downward, and then we would end up below average. So whether we predict above or below average depends on whether the correlation coefficient is positive or negative, and it also depends on whether we start out above average here or below average there. And it's best to make a picture and see which side you have to end up. So remember, in order to do regression, we must have a scatter plot that looks somewhat football-shaped. It turns out, in that case, not only can we compute a regression line, but we can actually also do normal approximation and say a little bit more about the y-values. For a given value of x, we can predict y simply by looking at the point that falls onto the line. But it turns out that the y-values of pairs which are near that x value actually follow the normal curve. So there's a normal curve for the y-values around that point. That means we can use normal approximation for those y-values. Remember, in order to do normal approximation, we need to know two things. We need to know the center and the scale of the normal curve in order to standardize. In the case of regression, these two numbers are given by the predicted value y hat, and the scale is given by the formula square root 1 minus r squared times the standard deviation of y. Let's do an example. Among the students who scored around 41 on the midterm, what percentage scored above 60 on the final? We already computed that the predicted value for a student who scores 41 on the midterm is 62.5. That means that the normal curve is centered at 62.5. So the percentage of students who scored above 60 on the final follows a normal curve that is centered at 62.5, and we want to figure out what percentage scored above 60. So that would be that shaded area. So now we simply standardize 60. We take 60, subtract off 62.5, and we divide by that formula over here. So that's square root 1 minus 0.67 squared times the standard deviation of the final exam scores, which is 11.8, and we find negative 0.29. So then we get a standard normal curve, and we have to figure out the area to the right of negative 0.29. And if you look up software, you find that this area is 61.4%. So the answer would be that among all the students who scored around 41 on the midterm, about 61% scored above 60 on the final exam. For each observation, we have an observed y-value and we have a predicted y-value. The difference between these two y-values is called the residual. Residuals are useful for checking whether the regression we are doing is appropriate. The main tool here is what's called the residual plot. It is simply a scatter plot of the residuals against the x-values. Here's an example. We have midterm scores on the horizontal axis and final scores on the vertical axis, and we also see the regression line through the scatter. Based on the regression line, we can compute the residuals, and those are shown on the right plot. What we are seeing in the residual plot is a scatter that doesn't show any structure. That's just the way we would like to have it. Here is another example where the residual plot shows that regression might not be appropriate. The left plot shows income versus education. And looking at the scatter, you can already tell that there's a lot of curvature in there. When we fit the regression and look at the residual plot, we see a pattern that shows a big curve. That's an indication that the scatter is not linear and a regression shouldn't be applied. However, not all is lost. It may still be possible to use regression for those data after we transform those data. For example, we could regress the square root of income or the logarithm of income on education. Sometimes that results in a linear pattern, and we can use regression on those transformed data. And then we would simply transform back the results. Here's another deviation of the linear scatter that a residual plot might catch. The scatter on the left-hand side is linear, so it roughly follows a line, but we see that it looks like fan shape. It opens up. And that also shows up in the residual plot. Such a scatter is called heteroscedastic. It simply means that the variability changes with the x-values. There's a little bit variability on the left side, but there's a lot of variability on the right-hand side. Those types of problems can oftentimes be fixed by transforming the y-values. However, it may turn out that that transformation on the y-values changes the scatter from linear to nonlinear. So we may have to do another transformation on the x-values to fix that. Here is an example from the 2000 presidential election in the United States. The scatter on the left-hand side shows the election results for each county in Florida, with the exception of Palm Beach County. In the 2000 presidential election, there was a third-party candidate, Pat Buchanan, who got a sizable proportion of the votes. The scatter shows the votes that Bush got versus those that Buchanan got. Just looking at the scatter, it seems that it's roughly linear, but when we look at the residual plot on the right-hand side, we see that it's heteroscedastic. However, if we take logs of both variables, we get a result that looks quite satisfactory. In particular, notice when looking at the residual plot, we get a band, a horizontal band, that doesn't show any structure. Now let's look at the same data, but including Palm Beach County. Palm Beach County is this one observation all the way up there. It's called an outlier because its y-value is very far away from the regression line. This is very clear by looking at the residual plot on the right-hand side. Again, we see Palm Beach County being very far above the horizontal line. Such outliers should be examined because they could represent an interesting phenomena. They could also simply represent a typo, in which case you may simply decide to remove it. This is another application of the residual plot because it makes it easy to spot those outliers. In fact, there was quite some controversy in the 2000 presidential election. The reason was that only Palm Beach County used the so-called butterfly ballot. And that ballot was suspected to confuse some voters to vote for Buchanan instead of the Democratic candidate Al Gore. That may explain why Buchanan got such a large number of votes only in Palm Beach County. Now you can see why regression is quite useful in all kinds of situations. For example, you could use the residual here to estimate how many votes Buchanan got in error. So far we only looked at y values which are outlying. An x value which is far from the mean of the x values is said to have high leverage. The reason why the word leverage is used is because such a point has the potential to cause a big change in the regression line. Let's look at this toy example plotted here. There are 4 points and 3 of them follow a roughly linear pattern, but the 4th one is quite a bit apart, and moreover it has a lot of leverage because it's far away in terms of x values. What would happen if we fit the regression without that point? Here's the regression line that we get if we omit this point here. We see that this one point has a big influence on the regression line. Such a point is called an influential point. Whether or not a point is influential can only be told by refitting the regression line without using that point. For such an analysis, the residual plot turns out to be not very helpful. The reason is that an influential point may have a residual which is quite small, so it doesn't show up in the residual plot. The reason why it's quite small is because the point is influential in the first place, so it pulls the regression line towards it. In fact, in this example, you see that the residual is quite small. Here are some other issues that you should keep in mind when doing regression. Remember, the main purpose of regression is to predict. Prediction should not be done at x values that are outside the range of the x values that were used for the regression. The reason for this is that oftentimes the linear relationship only holds for a certain range. We have no reason to suspect that it holds outside the range of the x values which we look at. Sometimes the data that are given to you actually come in terms of summaries, such as average of some other data. Those summaries are less variable than other observations, and the consequence is that correlations tend to overstate the strength of the relationship. Finally, most regression analyses report a number which is called R squared. This is simply the square of the correlation coefficient. The interpretation of R squared is that it gives the fraction of the variation in the y values that is explained by the regression line. So 1 minus R squared is the leftover variation that's left in the residuals. A high R squared means that the regression line does a good job in explaining a lot of the variation in the y values.
Traditional drug discovery is typically led by biologists, right? And so, in a program like liver cancer, that would, most typically entail someone who has spent their entire life studying the disease, and maybe they come up with some piece of biology or some mechanism that has the potential to be modulated, right, to help people with that disease. That type of research, can take decades. A lot of basic science, a lot of cognitive thinking. We replace a lot of that work with data science. We collect tons of data, more data than any human could possibly ingest and interpret on their own, process that information, and essentially come up with the same types of biological theories that you could apply, but all this happens in the course of a few minutes rather than the course of a few decades. The challenges we have have very little to do with technology choices and generating potential new drugs that have promise. More of our challenges are on it's an industry that has a lot of tradition behind it of having that classic lab coat scientists doing that work, And most of our work is helping to educate people that data science can actually make this difference, right? You don't need some prestigious professor who's been at it for 20 years. That kind of software can do the same thing, if not better. Like, that concept for people in this industry is a very tough one for them to deal with. And even just in the medical profession as a whole, there's been, I think, a lot of talk about what is the role of the clinician, right? What's the role of your doctor moving forward, and does that become one where they're doing less on decision support and software is doing that, and it's more about, you know, sort of that personal care perspective. Like, medical doctors don't like that, and they don't like that, and so that a lot of the work that we have to do is more around education and getting people comfortable and sitting down with prestigious scientists and telling them something that's a little out of their comfort zone. That's most of my challenges. It's not, strangely enough, it's not writing code. It's not solving technical issues in that regard.